Hello everyone. In this video I'm going to speak about a really interesting technique ver used widely nowadays, which is PCR, polymerase chain reaction. I'm going to speak about everything, the principle, the technique, the procedure, everything. So let's start by saying or by asking, what is PCR? PCR is a technique of DNA amplification. So, what is DNA amplification? So, let's say we have a DNA, we extract DNA from a human cell, and we are interested in a particular gene or a particular DNA sequence. Let's say we want to examine it, we want to analyze it, we want to clone it, we want to do anything with this sequence. So if we have one copy of this sequence, it's of course it's not enough to do anything, but we want to copy this sequence. We want a large or a huge number of copies of this particular sequence. In this case, we use PCR because PCR can give us millions of copies out of a particular sequence of DNA. And in a short time, in like 1 hour, we can get millions of copies of this particular gene or particular sequence. Now, how did the technique work? If I want to say one word about PCR, PCR is a thermocycle. What is a thermocycle? So I can say it's an increase and an increase and decrease in temperature. So we heat up and we cool down our mixture in order to produce multiple copies of DNA. So it's like this. So let's say this is temperature. We increase or we heat up the temperature, then we cool down, then we heat up again, then we cool down, and in a cyclic way, as you see here. So this, let's say this is one cycle, this is another cycle, this is the third cycle, and so on. So it's a thermocycle. Of course, this is very, very, very general. I'm going to speak about everything in details in few minutes. Okay, so generally or principally, PCR is a thermocycle. It depends mainly on temperature. Okay, so let's say this is our tube. We want to perform PCR in this tube. What should we have in this tube? So let's say PCR is DNA generation because it's a DNA synthesis. We're copying a particular sequence of DNA. First of all, of course, we should have the parent DNA. We should have our sequence of interest or the DNA sample. Okay? Now we need to copy this particular sequence. We want to synthesize new sequence of or new sequences of DNA. In this case, we need, of course, the DNA contents. And the DNA contents are the deoxynucleotide 3-phosphates, which are DNA contents, the cytosine, guanine, adenine, thymine. And the deoxynucleotide 3-phosphate looks like this. So we have the deoxyribose sugar, and we have the bases here, one base, adenine, thymine, guanine, cytosine, and we have 3-phosphates. So the generation of DNA will, or the synthesis of DNA will take place starting from these materials. Third, what should we have? So now I have the parent DNA. I have DNA contents in order to synthesize new DNA. I need something very important, which is the primer, in order to point on the particular sequence of DNA I want to copy. So the primer is very specific to the sequence or to the gene I want to copy. Primer is, I can say, it's a starter of DNA synthesis or DNA copying of the particular sequence. For PCR, I need a couple of primers, the forward and the reverse primers for the two complementary sequences of DNA. And also, you're going to see this in details in few minutes. But yeah, keep in mind, I need two primers, the forward and the reverse primer. And fourth, of course, I need something for DNA synthesis, very interesting, very important thing, which is DNA polymerase, of course. DNA polymerase in PCR should be heat resistant because, as I told you, PCR is a thermocycle, so it depends on heat, on temperature, and I need to increase the temperature in PCR, so I need the DNA polymerase to be heat resistant. There is one known PCR DNA polymerase, which is the Taq polymerase, but there are others, of course. But Taq polymerase is one very known polymerase for the PCR. Okay, so these are the four essential components I need to start a PCR. Now, what is the procedure? How does PCR work, or the steps? I told you PCR is a thermocycle. So the it's a thermocycle which is repeated several times. But in each thermocycle we have three steps of temperature changes. So in each cycle we change the temperature 3 times. So now I'm going to speak about the 3 steps of the thermocycle of the PCR cycle, which are first step is the denaturation of hydrogen bonds. Denaturation of the DNA. What does that mean? Saying we have this DNA, okay, this is double-stranded DNA, and we need to copy this particular sequence. Okay, this specific sequence. Of course, first of all, I should separate the 2 strands from each other. And as you know, there are hydrogen bonds here between the two, binding the 2 strands together. So I need to break these hydrogen bonds. How does it work? With heat, with temperature. So this is the denaturation of hydrogen bonds. So this is, let's say, the particular sequence I need to copy. So the 2 strands are bound together by hydrogen bonds, and to break the hydrogen bonds, I need heat. So I increase the temperature. I heat up my PCR mixture to between 94 and 98 degrees of Celsius for 20 to 30 seconds. Okay. After 20 to 30 seconds, the hydrogen bonds will be broken, so I will have 2 separated strands of DNA. Normally, the temperature here and the time depends on the GC content of the sequence. Why? Because as you see here, the guanine and cytosine bind together with 3 hydrogen bonds, while thymine and adenine bind together with 2 hydrogen bonds. So the GC bond is stronger than the TA bond, and because of this is that when I have higher GC content in my sequence, I need more time. I need like 30 seconds, and I need higher temperature in order to break the GC bonds. Okay, so this is the first step, the denaturation of hydrogen bonds. Now we have 2 separated strands. At the end of the first step, we have 2 separated strands of DNA. And then we go to the second step. In the second step, which is the annealing step, in the second step, so now we have 2 separated strands of DNA. Okay, so this is 5 prime, 3 prime, this is 3 prime, 5 prime. These 2 strands are complementary. In the annealing step, there is the hybridization of the primers with the DNA strands. So these are the primers. The primer normally is between 18 and 30 nucleotides. And the primers bind to the DNA or hybridize with the DNA exactly upward the sequence, the particular sequence, in the 3 prime side of the sequence. And why the primer always is deposited on the 3 prime side of the sequence? Because the synthesis of DNA takes place always from 5 prime to 3 prime. So we need the copied strand or the copied sequence synthesis to take place from 5' to 3'. Okay, so this is the forward, this is the reverse. I told you we have a couple of primer. The forward is for the first strand, the reverse is for the complementary strand. So the synthesis of DNA should take place from 5' to 3'. If you want to know why the synthesis of the DNA takes place always from 5' to 3', I explained this clearly in a previous video. You can find the link in the description of this video. So go to the other video and watch it. You can understand why the synthesis of DNA takes place always from 5' to 3'. But what you need to know at the moment is that DNA synthesis takes place always from 5' to 3', and because of this, the primer hybridizes with the DNA on the 3' side of the sequence, just upstream the sequence of interest. Now for this step, I need to cool down the temperature to between 50 and 65. If you remember in the first step, the temperature was between 94 and 98. Now the temperature is between 50 and 65 degrees of Celsius for 20 to 40 seconds. The temperature here depends on the primer. Why? Because the primer needs a particular temperature in order to hybridize with the DNA. So if the temperature is too low, if we have low temperature, the primer would not bind to the DNA. And if the we don't need the primer just to bind to the DNA. We need the primer to bind specifically to the upstream target. So we need higher than 60% match between the primer and the DNA sequence in order to be specific. We don't need the primer to bind here or there or there. We need the primer to bind specifically upstream the target. Okay? So we need higher than 60% of match, and because of this we cannot raise the temperature. So I told you, if we have cool temperature, the primer would not bind. And if we have a high temperature, the primer would bind non-specifically. The primer would bind here, for example, non-specifically. And we don't need this. So there is a rule of thumb, we can say. Normally for this step, we use a temperature which is 4 to 5 degrees lower than the melting temperature of the primer. So let's say we have a primer who has a melting temperature of 60. In this case, we use 55. Okay? Now, this is the annealing step. And the annealing step is nothing but the hybridization of the primer with the DNA sequence or with the DNA strand. Now, going to the third step. In the third step, the copying of DNA starts. So it's the elongation step. Here we have nucleotides, and the copying of DNA starts in this step. So here we have the primer. So let's say this is the sequence of the primer. I told you the primer is something between 18 and 30 nucleotides. The primer provides a hydroxyl group. This is the hydroxyl group needed to build up the new nucleotides. The hydroxyl group is on the 3' end of the primer. Yeah, again, you can find all these details in the previous video, whose the link is in the description of this video. So the hydroxyl group is needed to start the DNA synthesis from 5' to 3'. And of course, in this step, we need something very important, which is the DNA polymerase. And we need of course, a particular temperature, because PCR is all about temperature. So we need a temperature between 75 and 80 degrees of Celsius. We apply it for around 2 minutes. And then the polymerase will start to build the new copy of DNA. Okay? At the end of the elongation step, we will get two separated copies of the DNA, of the particular sequence of DNA we need. Of course, the 2 minutes are variable. They depend on the length of the sequence. So normally we need 1 minute for each 10 for each 1,000 bases, but yeah, it's somehow around 2 minutes. So at the end of this step, as I told you, we will have two separated copies of the particular sequence of DNA. So these are the three steps, and if we want to look at the thermocycle altogether, it looks like this. So this is the PCR thermocycle. So it starts by heating up the temperature, starting the DNA denaturation step. Then we cool down the temperature in order to, as I told you. So in the DNA denaturation, as I told you before, we separate the two strands of DNA from each other. And it's around, as I told you, it's around 94 to 98 degrees of Celsius. Then we cool down to the temperature for the primer annealing. We need around 50 to 65 degrees of Celsius. In this step, the primers hybridize with the DNA. The forward and the reverse primer hybridize to the DNA on the 3' side of the sequence. Then we heat up the temperature again for DNA extension, in which the new copy of DNA is synthesized, and the elongation of DNA takes place from 5' to 3' end. At the end of the DNA extension, it's the end of the first thermocycle. So thermocycle is 3 steps. This is the end of the thermocycle. Then we heat again. We have denaturation again. Primer here at the end of the second thermocycle. Okay, we can have as much thermocycles as we wish. So what I want to tell you is that at the end of the first thermocycle, we get 2 copies of the needed sequence. And then when we start with the second thermocycle, each one of these 2 will be copied to another 2. So at the end of the second thermocycle, we get 2 copies from each copy. So we get 4 copies at the end of the second thermocycle. So let's see how does it work. So we start with 1 copy, with 1 sequence. At the end of the first cycle, we have 2 sequences. At the end of the second cycle, we have 4, and then from 4 we have 8, and then from 8 we have 16, and so on. So it's like a cascade. Now there is so how many copies I will have at the end? The number of copies I will get at the end is 2 to the power n. N is the number of cycles. So let's have I can give you an example. Okay, so let's say we run the PCR for 1 hour. Okay, what would we get if we run the PCR for 1 hour? So let's say we run the PCR for 1 hour. So if one thermocycle takes around 2 to 3 minutes, okay, let's say it takes around 2 to 3 minutes, 3 minutes. So in 1 hour, I will have 20 cycles, and then the number of copies will be 2 to the power 20, which is 1,048,576. So imagine after 1 hour of PCR, I will get over 1 million of copies out of 1 copy, out of 1 copy of DNA, which is cool, which is pretty cool. With this 1,048,000, I can do whatever I want to do. I can clone the sequence, I can study it, I can analyze it. So let's say what applications of PCR do we have? We can do DNA isolation, we can isolate a particular gene, we can do DNA quantification, we can verify the presence of the gene. We can do gene testing in order to see if we have mutation, gene matching, the oncogenes. We can do viral and bacterial infection diagnosis like HIV, for example, or tuberculosis. We can use PCR for forensics, of course, because we can amplify the DNA we have, we can amplify it. So there are many, these are a sample of applications of PCR, but in reality, we have hundreds of applications of PCR. This is everything I wanted to tell you about PCR. I hope you understand this video. There will be another video about real-time PCR. So if you like the video, if you benefit from this video, like the video, share it with your friends, subscribe my channel because there will be always new videos. And thank you for watching. See you in the next video. Bye.